Welcome to Jerry's Gene Scene. Today I'm going to take a look at chromosome mapping. This is a technique that more than a century ago helped demonstrate that chromosomes play a role in inheritance. In previous videos I have shown you some important ratios. We need to remember that these are inferred ratios and our observed ratios may be somewhat different. Scientists have statistical methods to determine if the difference between the inferred and the observed are due to chance, or if there might be some other explanation for these differences. Now let's go back and look at our yellow-eyed, feathered antenna phantasmas. Through careful selection, we have pure-breeding yellow-eyed, feathered antenna, and we have white-eyed, straight antenna phantasmas. Now if we do an F1 cross, we find that 100% of the offspring have the phenotype yellow-eyed, feathered antenna. Yellow eyes and feathered antenna are dominant. In a previous video we learned about linked genes. We can establish using the Mendelian algorithms that there are two possible expected offspring outcomes from a test cross. Example 1, the genes are linked. We have two phenotypes, a test cross ratio of 1:1. In example 2, we have genes that are not linked. There are 4 phenotypes possible in a 1:1 to 1:1 ratio. Now let's look at the actual observed numbers. Out of 1,000 offspring, we get 480 dominant yellow-eyed, dominant feathered antenna, and 480 recessive white-eyed and recessive straight antenna. That appears to be a 1:1 ratio, except we also get 20 dominant yellow-eyed recessive straight antenna and 20 recessive white-eyed dominant and feathered antenna. How do we explain this? We learned that homologous pairs of chromosomes align during prophase 1 of meiosis. We also learned that some of these chromosomes cross over. Then, during anaphase 1 of meiosis, they move apart. When the gametes form, we have the possibility of 4 different combinations of linked genes. We can also imagine that there are fixed positions of the genes on the chromosome. When the number of offspring is large enough, the percentage of recombinants seems to be a reproducible number. Geneticists have used this number to help map the chromosomes using the percentage of recombinants as a mapping distance. I kept the mathematics and the statistics at a lower level to help understand the basic principles involved in chromosome mapping. Exam questions may even give an array of mapped distances and ask the student to determine the order of the genes on the chromosomes. If you found this video helpful, give it a thumbs up. And for a complete playlist on this topic, click on the video on the left. For more on Jerry's Gene Scene, hit the subscribe button on the right. I'm Jerry Ward. I'm at genomealberta.ca.